The process of gene expression varies not only during the different stages of life or throughout development, but also between different cell types. And so you can imagine that in order to exert different functions, a neuron or a nerve cell might need to express a very different set of proteins than a lung cell would, right? Or an eye cell versus a skin cell. And since every single cell contains the same genetic information or all of the same genes, it's the expression or which genes are transcribed and then translated into protein that actually varies between cell types. And so there has to be some type of regulation for which genes are expressed or turned on and which genes are silenced and turned off between different cells as well as in different life stages. And so I'm going to talk a little bit now about how gene expression is regulated at the level of DNA structure as well as at the level of transcription. And so the first way that gene expression can be regulated is by changing the nucleosome structure. And sometimes this is referred to as epigenetics, or the epigenome, which is the studies of how changing nucleosome structure changes gene expression. And so it's important to note that genes are only going to be expressed or turned on when the DNA is accessible to the transcription machinery, right? Because you need DNA as a template to make RNA. And so if RNA polymerase doesn't have access to that DNA template because that DNA is not accessible, their genes are not going to be expressed. And so under normal conditions, we already discussed sort of that normal nucleosome structure, that packing of DNA and wrapping around histone proteins. In this case here, where you have the double helical DNA wrapped tightly around histones, gene expression would be inhibited, right? Because the DNA is bound to histones and it's not accessible to the transcriptional machinery of RNA polymerase. But there are ways to modify how tightly wound your DNA is around the histone proteins to sort of adjust expression of genes. And so one modification that can be made to histone proteins that actually permanently will turn off gene expression is what's known as DNA methylation. And so methylation is basically just the addition of a methyl group. And if you remember back to the previous lecture, it's actually the tails that hang off of these histone proteins that will have the methyl group added to them. And so once DNA is methylated, it's actually irreversibly turned off. And so methylation patterns can give you an idea of which genes are never expressed in certain cell types or in certain individuals. And that is sort of unfortunate, right? Because once DNA is methylated, it's sort of turned off forever, and the genes that are in this DNA wrapped around a methylated histone protein can never be expressed. What's interesting about DNA methylation is it's actually inherited from person to person, from parents to progeny. And so if DNA is methylated and that gene's expression is turned off in a parent, it will also be subsequently turned off in the offspring. Also, interestingly, 90% of our genes are actually subject to DNA methylation, and about 10% of them can actually have their expression modified and turned on and off. So a lot of the DNA in our cells is actually kind of not expressing genes ever, right, because it's methylated. And so there are two other types of modifications that can be made to the histone tails that actually provide some reversible inhibition and activation. And those modifications are the addition and then the removal of acetyl groups from the histone tails. So once again, as I said, it's the tails of the histone proteins that get an acetyl group either added to them or removed from them through histone acetylation and deacetylation. And what histone acetylation does is that acetyl group can actually push these two proteins apart and open up or unwind the DNA in between them and allow gene expression to be turned on. Because those genes are unwound from their histones, they're now accessible to RNA polymerase, and so expression can happen. right, and be turned on. And then there's sort of the reverse is also possible, and that acetyl group that may have originally turned on gene expression here can actually be removed, or that histone tail can be deacetylated, and the DNA can be rewound and packed tightly again to turn gene expression off or inhibit it, right? And so all of these modifications really just change how accessible the DNA is. Methylation makes the DNA inaccessible, and it does so irreversibly by binding to histone tails here. Acetylation makes the DNA more accessible and unwinds it from the histone proteins and allows gene expression to turn on. And deacetylation removes that acetyl group, rewinds the DNA, and turns gene expression off again. And so these changes to the nucleosome structure are one major way that gene expression is regulated. There are also control elements within the DNA itself that act to kind of turn gene expression on and off. So not only does access to the DNA matter, but the sequences around the DNA also matter. And so there's a sequence in the DNA that does not code for a protein, but it does signal an increase in transcription, and that DNA sequence is what's known as an enhancer sequence. And there is a particular protein called an activator protein, which is a transcription factor that will bind to an enhancer sequence and promote transcription. And so you can see that happening here, right? The activator protein, or this transcription factor, can actually come in and bind to the enhancer region and recruit RNA polymerase to activate transcription. And the difference between the enhancer and the activator is, once again, just the enhancer is the DNA sequence that the activator protein binds to. So enhancers are sequences of DNA and activators are proteins. And there is also sort of a way to turn genes off in the same way, with a sequence in the DNA that decreases transcription, known as a silencer. And so there's a silencer sequence here, and there's a specific transcription factor protein called a repressor that binds to this silencer sequence, stops RNA polymerase from being able to bind, and therefore turns off transcription. And so these control elements, both enhancers and silencers, exist in the DNA upstream of the promoter to either basically increase or decrease transcription. And in order to exert that increase or decrease on transcription, they require activator and repressor proteins, or transcription factors. And finally, gene expression, or the ultimate kind of phenotypic output of protein from an RNA transcript, can also be regulated by what's known as alternative splicing. And so when we're talking about nucleosome structure changes and histone modifications, or we're talking about control elements, we're looking at regulation of gene expression at the level of transcription, right? So changing the nucleosome structure either allows or disallows transcription. If we have a repressor or activator protein that either increases or decreases the level of transcription. But this alternative splicing acts between transcription and translation. So it's sort of a post-transcriptional regulation of gene expression. And we already discussed how the RNA transcripts are a series of exons and introns that must be spliced before they leave the nucleus to be translated. And what's interesting is that this splicing does not have to include every exon in the final transcript that gets out to the ribosome. And what that means is that there's kind of alternative splicings of the same transcript that can result in different proteins. So for example, on the left here, protein A contains exon 1, 2, and 3, whereas protein B contains exons 1, 2, and 4. And there might be another protein that could contain just exon 2, or just exon 3, or all 4 exons. And this idea of having one RNA transcript that can be spliced in different ways to create different proteins is another way that we can sort of regulate gene expression, right? So by changing how this mRNA is spliced, we can either turn certain proteins on and favor others, right? Or we could make these two proteins and not others. And this gives some control over the phenotype or the phenotypic output of gene expression as well.